Hello, everyone, and welcome back to Psychedelic Vantage. Today, I'm speaking with Dr. Sam Clark, founder and CEO of Terran Biosciences. By way of background, Sam completed his undergraduate degree in neuroscience at MIT and received his MD and PhD at Columbia University. Sam founded Terran in 2017 with the mission to transform the lives of patients living with neurological and psychiatric illnesses. We have seen a string of some very significant announcements come out from Terran this year, and I am excited for Sam to share more about the work Terran is doing. The goal here is to learn more about Terran's strategies to bring their drugs to market, to learn more about the company's recent announcement, the development of TerXT, a combination of novel prodrugs of xanomeline and trospium designed to be long-acting treatments for schizophrenia. And we will also get to learn more about how Terran is combining psychedelics with highly selective serotonin 2A blockers in order to retain the therapeutic benefits of psychedelics without the associated trip for neurodegenerative disease as well as depression. Terran is working on many other different compounds along with novel technologies to help patients. And if we have time, we will cover these other programs as well. Sam, thank you so much for taking the time to come on the channel today. How are you doing?
I'm great. Thanks for having me on.
Absolutely. So to get started, can you give us a little bit of a background on Terran, a little bit about how the company was founded, the company's mission, and also corporate structure?
Yeah. So I founded Terran based on a number of life experiences. Growing up, there were a number of family members and friends with severe mental illnesses, particularly bipolar disorder. And then we also had family members with neurodegenerative diseases. My grandmother died from Alzheimer's disease. And so I wanted to make a difference in neuropsychiatric diseases. And the other issue was just seeing how the medications that people had access to had a lot of side effects. They were slow to work. They were using mechanisms that had been invented particularly like 40 years ago, in some cases longer. And just the other issue is the cost of medication. Some people wouldn't be able to afford branded, medication here in the U.S., on older generic meds. So it's really eye-opening for me. And so that's why when I went to college, I was studying neuroscience, went to medical school, focused on neuro, did my PhD. And that's where I really wanted to develop new drugs. At the time, I wanted to be a doctor, but then I said, the problem is it's just the slow pace of drug development and the lack of options. And so I founded Terran to address that. I said to develop new treatments for people with neurological and psychiatric conditions. And I found it in a way where I wanted to build kind of a different corporate structure in order to really transform the space. That was mainly because at the time, I was looking at all of the companies in Silicon Valley and how fast they would move, the tech companies. And then I was looking at biotechs. I was like, the pace of research and development and discovery is so slow. Look, in general, we all know it takes a long time to get a drug past the FDA. But apart from that, apart from the regulatory and normal delay structure, the issue I was looking at is that based on the way these companies are structured in this kind of pyramidal top-down corporate hierarchy, it just takes a long time for things to move up and down the chain. It takes a while for new discoveries to be made, and most companies are only focused on doing one thing, like one therapeutic, for example, and it leaves a lot of risk if that fails. And so the whole structure I thought needed to be changed. So I founded Terran structured as a tech company with a flat corporate structure. So we do things quite differently here. We have a small, highly trained team of core operators. And then we bring on world experts to solve problems that people previously thought impossible. So we think up I have a number of ideas, and then I'll build these teams of world experts around it. And then the operators in the core team help keep everything organized between the teams and within the teams. And in that, we have access to some of the world's top talent. We're able to address problems that people previously thought were not possible to address. And we move very, very quickly.
Thank you for that overview, Sam. And I would just like to ask you, and correct me if I'm wrong here, I believe Terran's initial focus right from the outset was schizophrenia. Um, I'm curious, what year did Terran start getting interested in the psychedelic medicine space?
So we didn't actually get into psychedelics until around almost 2021
Oh,
was
okay.
when. And so the what happened was we were very much in the traditional schizophrenia space. And I'll tell you about one of our lead assets there, idazoxan, and also
Yep.
our new xanomeline trospium prodrug combination a little later. But
Yes.
the issue was that while I was very interested in psychedelics and had done a lot of research on psychedelics before, we didn't see a unique angle by which Terran could really contribute to the space at the time. So we had
Mm,
always held off getting into the space. But it wasn't until we came across a number of breakthroughs at academic labs where
mm,
they were showing that you can take the psychedelic, remove the trip by combining it with a selective serotonin receptor blocker, and still get the therapeutic effect in animals. And that we said, wow, this could be huge because it could increase patient access. If people didn't have to take the trip, it could greatly increase the number of people who had access, and it could greatly
mm,
reduce the cost of the drugs. And those are two critical components of our mission, which is to increase access, give people access to unique, right-sized solutions, and without at an affordable price point. So we got into psychedelics around that, licensed the selective blockers from Sanofi.
mm,
Uh, then we spun up large-scale GMP manufacturing. Now, Terran is one of the largest manufacturers of psychedelics in the world. We manufacture GMP LSD, MDMA, psilocybin, and many other psychedelics. And so the idea is that after we spun up all of that infrastructure to really develop that program, we realized there was a lot of other unmet medical need in the psychedelic space that wasn't being addressed, and some concerning developments as well. I've always been against the idea of patients, having drugs that are new but they can't necessarily afford. And we saw that what appeared to be potential monopolies set up around certain psychedelics in regards to salts and polymorphs. And so we said, we can use our expertise to break through those monopolies by designing new salts and polymorphs that will enable us to do 505(b)(2) applications
mm.
on a wide variety of psychedelics. It wasn't the goal of getting into psychedelics to do 505(b)(2s, but once we had large scale manufacturing ongoing and we're producing, multiple GMP runs and we have all this excess psychedelic, we said, well, it's a small leap to also use these to do 505(b)(2s and make sure that patients are never overcharged for their medications.
Mm.
So wherever there are these monopolies being set up, we've been able to get in there and break through those IP barriers quite effectively.
Very interesting, Sam. Thank you. And yeah, it's been very interesting to see the announcements made by Terran this year, and really made me look at the space a lot differently through these announcements that have come out. So let's speak about the most recent announcement from Terran. Can you explain the significance of Terran developing TerXT with plans of pursuing the 505(b)(2) pathway to bring this drug to market?
Sure. So TerXT is a combination of novel prodrugs of xanomeline and trospium. Now, in order to fully understand what's going on here, I think we're going to need some background information.
Mhm.
So let's cover what is a prodrug. So a
Yep.
prodrug is when you have an active compound and you attach a side chain or another what's called moiety to it, and that improves the pharmacokinetics of the original compound. But once it reaches the bloodstream, the side chain is removed and the active ingredient reaches the brain. And you think about it like this: if this is the molecule, you put the side chain on it, it gets in the bloodstream more effectively, or it fixes pharmacokinetic problems with the drug. It could increase stability. It could make the drug better absorbed in the colon. It could make the drug better for a long-acting injectable. But once it's in the bloodstream, this is gone.
Mm.
And that means you have the original active ingredient in the bloodstream. And now you're in the best of both worlds. the patent office recognizes that this is a completely new composition of matter molecule. It's a new molecule completely.
Okay.
The FDA recognizes that the active ingredient is the original compound. And so you're eligible to use the 505(b)(2) pathway usually. Now, let's cover like what has been done there for antipsychotics. This is a classic play for antipsychotics. So if you think about it, most of the best antipsychotics to date have all already been made into prodrugs. And those prodrugs have already been FDA cleared on, and many times on the 505(b)(2) path. So for example, the drug Abilify, that's aripiprazole, that was one of the most effective antipsychotics to date. That drug was approved in around 2002. 2002. So 22 years ago. So back in 2002. But the first, the long-acting injectables came, over 10 years later. And the first one was as a prodrug, Abilify Laroxel. And that was the 505(b)(2) application. And for example, with elaperidone and other long-acting antipsychotics, that's Invega Sustenna, Maintena, Trinza, and now once every 6 months, Invega Hafyera, that's a prodrug as well, and 505(b)(2). So the idea is that you can really improve antipsychotics via the prodrug approach. It's a classic approach. It's been done many times before. And you usually take the 505(b)(2) path. Now let's explain what is the 505(b)(2) path.
Yeah.
So when you typically get a drug approved, you take the 505(b)(1) path. Now the 505(b)(1) path means you have to do 2 phase 3 trials at least. So, most companies will run a third in parallel or a trial in Europe. You have to expose a large number of patients. You have to have a safety database over a year. There's a lot of requirements, and it takes a long time, and it can cost hundreds of millions of dollars. But once the FDA is satisfied they understand the drug and the blood levels of that drug required for efficacy, that's when you can take the 505(b)(2) pathway. And the 505(b)(2) pathway allows you to reference all of the data from the first company that submitted the drug, whether or not they allow you to reference it. And we'll get to the rules around that in a minute. It's very interesting. So you don't have to repeat the study, the efficacy studies, and those can be, that'd be wasteful spending. That would be exposing people who don't need to be exposed. So you don't have to repeat that. You have to show the FDA that you can, number one, achieve pharmacokinetic equivalence in a bioequivalency study. And that can be a relatively small trial. And then you have to show sometimes bridging safety. So you have pharmacokinetic bridging and you have bridging safety. And those are usually both pretty easy because the main compound's already shown to be safe. So that can be very quick, very cheap compared to a 505(b)(1) and put another drug on the market. Now, let's kind of go into another thing people think about called marketing exclusivity. When that 505(b)(1) hits the market, they get 5 years of exclusivity to their data. Now, that is only exclusivity against other people referencing it. Another group can submit a whole nother 505(b)(1) for the same drug, same indication at the same time. But so the exclusivity does not block other 505(b)(1s, but who's going to realistically do for 2 phase 3s when there's already a drug on the market with the same indication, same molecule? Nobody. So, or if they wanted to, no one's going to fund it. So in reality, once someone is on the market, people just wait the extra years, and then they can do the 505(b)(2) application after the exclusivity period expires, approximately 5.5 years later. Now, here's where it gets a bit interesting. Most companies, when they launch, have a composition of matter patent on the original molecule, which means it doesn't matter when the exclusivity expires. The patent doesn't expire for, on average, it can be 20 years later. And since it doesn't expire for about 20 years, they have exclusivity far beyond the FDA data exclusivity. But in some cases, for example, many psychedelics, they don't have that level of exclusivity. And that is great for patients in that it enables other companies to come in, use the 505(b)(2) pathway to put generics on the market or to put other branded products that can reach bioequivalency with improved benefits. So you have a couple options. Let's go over them. 505(b)(2), number one, you could put the same molecule, same salt on the market if you can get around any patents there. That's a generic. It'd be the same thing. You can switch the salt and switch up some aspects like make a prodrug or deuterate the molecule. Many people forget that deuterated molecules are also eligible for the 505(b)(2) pathway.
Mm.
And then you show bioequivalence to the original molecule, and then you get your approval, and now you have another branded drug on the market with different pharmacokinetics, often improved from the original. That still produces competition in the market, which is what we like to see. That keeps the price low so patients have access. And so following all of that, the 505(b)(2) is great for patients. Now, there are a few techniques people are using, companies are using, to stop the entry and to extend their exclusivity beyond what they should be getting. Now, here at Terran, we're not anti-patent. If you have a patent on something, and people need to get rewarded for their innovation, so we're happy with patents. But there are techniques people are using that we don't agree with. And for example, one of them is the 30-month stay on the 505(b)(2). Now, let's really understand what is that. What that means is that when you get your drug approved, you list in the Orange Book the patents that protect your drug. So let's say you get a form of, psilocybin approved. Mm-hmm. And let's say you only have a or let's say it's LSD, and you only have a manufacturing patent on that LSD covering one salt. Let's say it's the tartrate salt. But the problem there is that if another group is developing LSD tartrate, you can file for the 30-month exemption, 30-month stay, and say, oh, I think they're infringing my salt. Well, then what it does is the FDA puts a hold on the data access, and it goes to the courts to decide if there's an infringement. Now, they may decide there's no infringement, but there's no, it wasn't a penalty for that group filing when no infringement occurred. There could be sanctions if you prove they did it maliciously, but it's very hard to do. And so the idea is that people are almost incentivized to file those stays no matter what. So that's why we develop new salts and polymorphs, to break through those monopolies and break through 30-month stays, because there won't be any 30-month stay if you're not even developing the same salt of the drug. It would be very hard to infringe any patent there. And so in that sense, because most of these psychedelics are dependent on salt and polymorph secondary patents, so in that sense, Terran files a lot of secondary patents that have been granted that will enable us to use the 505(b)(2)s on the excess GMP we manufacture from LSD, MDMA, R-MDMA, for example, and many other compounds, psilocybin, to make sure patients get cheap, affordable medications that are not delayed.
Thank you so much for that really detailed overview, Sam. It's very helpful for myself and also the viewers.
And we're going to get to TerraXT next. I just wanted to give that background first on 505(b).
Okay, yeah, go ahead, speak about TerraXT.
Great. So TerraXT is another one of those unique situations where we say, okay, how can we get a modern technology affordable drug to patients? Now, this was interesting. We had watched the development of the current twice daily oral xanomeline trospium combination that has gone through phase 3 clinical trials. It's been submitted to the FDA, and that was recently acquired by Bristol Myers. Now, that current once daily formulation is protected by a large number of patents around that specific compound, xanomeline, and a specific compound, trospium. However, it's twice daily. That's a twice daily oral. Now, there's something that's gone on in big pharma for a long time that's always frustrated us, and that is the life cycle management strategy that I've always been very critical of. That strategy we've seen, for example, with Abilify, when we just saw last year that the once monthly injectable Abilify went off patent, and the group Otsuka was set to lose quite a large amount of revenue from that. And the once every 2 months was then put on the market to replace it, or just got FDA clearance. It will replace the once monthly. Now, the question is, it's interesting that big pharma usually replaces a drug that goes off patent with a better version of that drug on patent. And they do that primarily when they lose the patent protection on the first drug. Similarly, Abilify was when it was going off patent when we got the first long-acting injectables, and from other companies that were able to get that in there. Now, the life cycle management, basically, they don't cannibalize their sales. So there's not a big incentive to improve the molecule before it goes off patent. Now, you might ask yourself, well, here we are in 2024. Did we just gain the ability to make once every 2-month injections? No. Invega Hafyera is once every 6 months. Invega Trinza is once every 3 months. But the idea is that patents expire, and then when the original form goes generic, and the doctor says, oh, let's go to the generic, suddenly a no branded one gets approved. Oh, well, this one's better. Stay on the branded, more expensive medication with a whole host of blocking secondary patents so nobody else can put another one on. And so we said, well, interestingly, if you look at some of the life cycle management strategies, once monthly injectables and once daily improvements to other antipsychotics, those just roll out very slowly. And I have been frustrated, since way before I founded the company, by the idea of patients not having access to cheap, affordable meds and the best technology available. So here at Terran, we said, well, look, we've already made major breakthroughs in the prodrug space. We made the world's first truly orally active DMT tested in animal models, what doesn't need an MAO inhibitor as a prodrug. We did it with 5-methoxy. We made long-acting MDMA as a prodrug. We prodrug new prodrugs of psilocin, basically new psilocybin type prodrug. Psilocybin is itself a prodrug of psilocin. And as such, and many others, we prodrugged, all sorts of other molecules, 2CB and LSD, and a whole bunch of other stuff, 2-bromo LSD. And so the thing is, we said, look, we have this twice daily getting approved. There are no patents that we're aware of that block prodrugs. We will build the prodrugs the same as has been built for all the other antipsychotics I've mentioned. Invega, haldol-decanoate, fluphenazine decanoate, Abilify Laroxal. We can make the prodrugs now. I mean, we could theoretically think that maybe Big Pharma will make them eventually, but the molecule has a lot of pharmacokinetic limitations. Xanomeline is invented in 1992. Trospium is invented in 1960. We're talking about 2 old, no composition of matter left molecules. I'm not saying the combination of the 2 is old, but the molecules themselves are old. So we said, let's give it a modern upgrade. We will use all of our technology here at Terran to improve these molecules to the best possible prodrug form they can be made into. And then we will make a fixed-dose combination of the 2 molecules and get those to patients quickly. And we'll take the 505(b)(2) path if possible. We'll meet with the FDA on that. Same as has been done for just about all the other antipsychotics. And so that's what we did. And we developed those prodrugs. It took 2 years to do. We did 365 animal studies. We did over 260 in vitro additional studies. There's more than 500 studies done. We had at one point over 200 full-time chemists working on the program, as well as teams of pharmacokinetic modelers. We built a whole population PK database for xanomeline and trospium. We developed the LAI PK model, the long-acting injectable, the once daily oral. We had teams on our CMC team scaling xanomeline up to large-scale manufacturing so that we'd be ready to go as well on the large-scale CMC side. So this was a multidisciplinary approach. Then we brought on some of the teams of people who had developed some of the best long-acting injectables in the past, and we brought them on to help us design our long-acting injectable plans as well. And so with all of that tech, we came out with amazing prodrugs. The patent for the composition of matter, the claims were recently allowed by the patent office, and we expect to move that to grant now. And we are very excited about this program because we believe this will potentially produce, in our opinion, pharmacokinetic benefits. We think we're going to get the once every several month injectable, multi-month duration, and a nice, good, long-acting once daily drug with an improved version. So you can fix the absorption problems present with trospium in the original.
I see. Thank you, Sam. And I would like to ask, how or why, how is Terran, I guess, able to accomplish this? Like, why aren't we seeing a bunch of other companies take the approach that you are? I guess with, I think you mentioned Otsuka waiting like 15 years to create a better, improved version. And you guys are getting to it, it seems, earlier with the drug that Bristol Myers is working on. Like, yeah, so how is Terran able to do this? Why Terran?
Well, I mean, in general, big pharma does typically move a lot slower than they used to on innovation because they usually follow now the merger acquisition model. It used to be, if you went back, more than 20 years back in the 90s, before it was all the innovation was really happening in big pharma. There weren't all these small pharma companies. It's a different model
Okay.
now. And now big pharma buys them up. But at the same time, then the question is, well, why aren't there all these other companies doing what we're doing? Well,
Yeah.
it's a couple things. Number one, it goes back to that corporate structure. Companies are structured and set up with their investors to really just do one thing usually. And if you get a bigger platform company like a Cerevel or Numora, they do a few things. They have a pipeline. But in our sense, we do have a lot of flexibility. And that we wield that flexibility by constantly searching for ways where we can improve patient access, break down barriers, and really get in there and break through potential monopolies or patent thickets that have been set up. And we're able to then move very quickly because of the lean structure here at Terran, where we're able to say, okay, we have a problem. It needs to be solved. Let's find who in the world can solve it. For example, when we set out to make prodrugs of xanomeline, which you can see behind me, and this is inspired by the xanomeline structure. But the idea is that initially, the chemist said, well, you can't really do it because the nitrogen, already has a methyl group there. But we did figure out a way to do it through quaternization. And there's some strategies we use that are not easy to do. And we had to pull on ultimately a team of 6 of some of the world's expert medicinal chemists, a whole team
Yeah.
to really design these structures. We designed more than 10,000 prodrugs for xanomeline. We made more than 500, and so, for example, one of the lead chemists said, in my 9 years at Big Pharma, we only made 300 prodrugs, and here at Terra now, and just on this one program, we made more than 500. So
Wow.
it's really exciting. And it was a multinational approach with groups working across Europe, Asia, United States, and others. It was really in the UK. It was a big, massive undertaking to get this done. But we're very excited because we think that this will represent a big breakthrough for patients.
Very exciting. And yeah, I guess no one should think that any of this was easy. I remember I looked on the website for TerXT, 40,000 hours of medicinal chemistry, hundreds of different chemists involved. Um, I guess before we move on to the psychedelic space, could you touch a bit about your idazoxan XR program? I know this was also another area where Terran broke grounds with medicinal chemistry.
Yes, so this is another one of our earliest assets. So the story here was that back in the 1990s, a company in France, Pierre Fabre, was developing a compound called idazoxan. This was the first ever selective alpha-2 antagonist. And there still exists no FDA-approved selective alpha-2 antagonists for any indication. And none approved in Europe or anywhere else in the world. So this was safe, and they were testing it in phase 3. So hundreds of people already dosed, more than 500, for Parkinson's tremor. Now, it wasn't very effective as monotherapy for Parkinson's tremor. But at the same time, there was a partnership at the National Institute for Mental Health in the United States. They used it in phase 2 trials in patients with schizophrenia. And what they found was really remarkable. They found that when you added it on to any existing antipsychotic regimen, it would double the efficacy of the baseline antipsychotic, up to double. It would produce an enormous benefit on top of the baseline antipsychotic. And so they wanted to advance that. However, the problem was the drug had such a short half-life, it had to be given at least 3 times a day in the big phase 3 trials. And Pierre Fabre said, okay, we can't fix this. And they say it's not really commercially viable anyway at a 3 times daily drug. So manufacturing was stopped, and they largely exited neuro at the time. Now, this was unfortunate, as patients and the doctor who ran the trial, who's now actually an advisor on Terran, an investor in Terran, said patients and their families were calling, begging to be put back on that drug, but manufacturing had stopped and there was no way to get access again. So we learned about this drug through our some of our SAB members, our science advisory board, and many of them had talked about the promise they had seen with this drug many years ago. And so what we did was we said, okay, we went to France, we met with Pierre Fabre. They gave us the rights to the drug. We then built a team of world experts to try to make a once daily form. Now, initially, it proved very difficult. So then we rebuilt the team and pulled on someone who had almost more long-acting once daily formulations than anyone ever in terms of FDA approved. And he had worked on some of the most difficult once daily formulations of all time. And he was actually about to retire, but he said, okay, I'll take this on, basically one last project before I retire. Brought it on and was able to create the once daily of the drug. So we went to the FDA, got the green light to go forward, put it into a big pharmacokinetic bridging phase 1. But as you know, it's already been in hundreds of people, but we wanted to show that we can do it. We brought 5 forms into that phase 1. And of the 5, they all showed a great once daily profile, but we found one that was better than all of them and chose that and are taking that forward. We then hope to advance that back into trials with schizophrenia. So that clinical trial is going very well, the bridging phase 1, and then we want to go back into adjunctive schizophrenia after. So that really is a key clinical stage program at Terran. And, along those lines, we're going to hope to advance TerraXT also into the clinic shortly.
Very, very cool. And what's so interesting about this is feels like such a unique opportunity here with the drug already showing prior efficacy, and then Terran was able to create a once daily pill. So really cool. I'm excited to see what happens with this program in the future. So I guess taking it to the psychedelic medicine space here, my understanding is that it was once thought impossible to create new salts and polymorphs of psilocybin. I guess, can you explain the significance of creating the first new forms of new salts and polymorphs of psilocybin, and what went into this?
Well, it's when someone says it can't be done, at Terran, we see that as a challenge to do it. And so that is exactly what they told us when we set off to make the new salts. They said, well, it's a Zwitter ion, and Zwitter ions don't form salts. And that's just the general accepted rule. And I said, well, we're going to figure out if that's true, because if they can form salts, we're going to make them form salts. And so it was over a year of really intense solid state chemistry, where we also brought on some of the world's experts in solid state chemistry, some of the best labs in solid state chemistry. And we did indeed create the world's first new salts, or first salts of psilocybin hydrochloride, psilocybin adesylate, psilocybin mesylate. The patents were granted for composition of matter of those salts, for the method of use, depression, and others, and pharmaceutical formulations. And with those salts and polymorphs, we are very excited to put 505(b)(2) applications of psilocybin on the market 5 years after whoever gets psilocybin approved. Or let me rephrase that. Also, of whoever gets a psilocin drug approved, because psilocybin is a prodrug of psilocin. The active ingredient isn't psilocybin, it's psilocin. And so for 505(b)(2s, you'd be arguing to the FDA that you reach effective psilocin blood levels, which means that our psilocybin salts and polymorphs should also be viable in 505(b)(2s against any psilocin products on the market. And potentially against deuterated forms as well, because if the deuterated form is ultimately forming deuterated psilocin. So we'll see. But it could go well beyond psilocybin, but the goal is it's going to go on the market we're going to submit that about 5 years after whoever gets approved. We're going to put that on the market, and it's going to lower costs and increase affordability for patients. And it's a side project for Terran at this point because, we manufacture bulk already for our fixed dose combination products. So this is kind of a bonus, but it's a bonus for patients. And we repeated that bonus with MDMA. We made new salts, polymers of LSD, 2CB, methylone, and many other compounds. We made it with ibogaine derivatives. We did it with additional DMT derivatives. And we have also, to shore up the 505(b)(2) program, we made prodrugs I mentioned earlier of DMT, MDMA, psilocin. Those prodrugs can also be potentially 505(b)(2) eligible. And we have deuterated forms. There are cases where there are new molecules being developed and that Terran then made the deuterated form. The reason why we made that deuterated form is that if the deuterated form is not covered in the original patent on the original molecule, then it's not blocked. Our deuterated form is not blocked. And if our deuterated form is not blocked, then you can guarantee that patients are going to have access to our cheaper, affordable medication to increase competition in the market 5 years after whoever gets that non-deuterated form approved. And that goes beyond, the psychedelics I mentioned into all those other, more niche and unique compounds. At the end of the day, you really have to ask yourself, who benefits here, and it's the patient, and then ultimately who could lose from Terran's strategy, and that is potential big pharma companies if they bought out the molecule, and potentially some of the big venture groups funding some of the other companies. But those big venture groups have plenty of money in the bank. It's not gonna really hit the bottom line. There's many, many billions in reserve, and those big pharmas have a lot of money too. So in this case, we are very excited to push forward to help the patient, because ultimately our only motivation here, the only final person we really care about there in that chain is the patient.
Thank you, Sam, for explaining what Terran is doing in this area with creating new polymorphs and salts of psychedelics. And what it could mean for patients and with the 505(b)(2) pathway. Before we get to the next question, I would like to show a video that Terran created in order to show how they are looking at psychedelic drug development.
Psychedelic compounds are currently under development for patients with neurological and psychiatric illnesses. Clinical trials of psilocybin have shown promising efficacy in patients with treatment-resistant depression. However, psychedelic therapy often requires a supervised multi-hour psychedelic trip and comes with the risk of a bad trip, and thus may only reach a very limited number of patients as second or third-line therapy. Terran is developing therapies combining psychedelics with serotonin receptor blockers that may have the potential to deliver the therapeutic benefits of psychedelic medicines without the associated hallucinogenic trip. This approach could increase the potential for broader clinical use of psychedelics and could expand access for many patients, including those with major depressive disorder, anxiety, and neurodegenerative diseases. Terran's non-hallucinogenic psychedelic therapeutics may have the potential to become first-line take-home therapies with superior safety, efficacy, and more rapid onset compared to standard antidepressants. In parallel, Terran is developing new, improved forms of classic psychedelics for other indications where the trip is thought to be an essential part of the therapeutic benefit, such as PTSD. Additionally, Terran is developing receptor-blocking compounds that may be able to end psychedelic experiences or tailor them to a specific duration. These compounds have previously been tested in over 15,000 patients across a variety of indications. Terran has formed partnerships with universities and clinics and is providing psychedelic compounds to researchers globally. With our unique approach, Terran Biosciences is well-positioned to transform the treatment paradigm for patients suffering from neuropsychiatric illnesses.
We are seeing many companies go about creating non-hallucinogenic versions of psychedelics that agonize the 5-HT2A receptor in order to treat a variety of psychiatric disorders. Terran is taking a different approach in regard to skipping the trip by combining psychedelics with highly selective serotonin 2A antagonists in order to retain the therapeutic benefits of psychedelics without the associated hallucinogenic effects. Can you please share to the viewers what inspired Terran to take this route, and also speak about the fMRI results you saw in humans when looking at this drug combination?
Absolutely. So first, let me clarify, there's two things we do. We have big libraries of preclinical psychoplastogens. Those are compounds, as you described, similar to what other company strategies have taken, where you modify the molecule so that itself has no hallucinogenic effect in animals. In some cases, we have deuterated forms of other existing psychoplastogens with the goal of doing that 505(b)(2) to increase patient access. And in many cases, we have our own entirely new libraries that we've designed that we think are better. So we have that already. But we always saw that as a very risky proposition because, yes, they might work in animals, but as we know, scientists have already cured all diseases known to mouse kind. It doesn't always translate to humans. So, we said, well, what do we know actually does already work in humans? The classic psychedelics. So if the classic psychedelics already work in humans, and those classic psychedelics hit a whole variety of receptors, and we know already that only one receptor is responsible for the trip, the serotonin 2A receptor, then the idea is if we can block just that receptor, but allow the psychedelic to hit everything else, we believe it'll maintain the antidepressant effect and thus help us obtain our mission at Terran, which is by doing so, helping to put a non-psychedelic compound on the market. This is something that could potentially be a take-home medication, not require the 6 to 8 hour therapy experience or just general trip required to be supervised by a doctor that increases the cost of medication, reduces patient access. This could greatly increase patient access and reduce cost. Now, we're not anti-trip at Terran. Perfectly happy if there are, we do make psychedelics with the trip as well. But this was a unique opportunity to also go that extra step for more access for patients. And so I can show you the data we did. So as I mentioned before, there's a lot of preclinical data in animals already that this would work. But when we say, okay, let's see if there's any human data. So this was an MRI analysis study that we did with in collaboration with a team at Yale and analyzing this MRI data set. And let me, I presented some of this at Wonderland. Let me show you kind of in detail what we're looking at here. So this was a study done in Europe where patients would come in and at baseline, they would get 2 placebo pills, multiple functional MRI scans, and then the trip would be assessed at multiple time points of the 5-ADC scale. Now, then, wait a minute. Oh, sorry, we're going the wrong way. There we go. All right. Then they would get, and this is crossover, so the order is all different. LSD, 1 placebo, 1 LSD. And, you get the MRI and you get a big trip. A classic LSD trip, 100 micrograms. Then you'd have the 5-HT2A block. So you get a 5-HT2A antagonist plus LSD, multiple scans, and no trip. So LSD did block the trip in these people. The 2A antagonist blocked the effects of LSD. But what was really remarkable is what we found when we analyzed the data. And that is that the functional network assignment of the blocker plus placebo actually shows a complete functional rewiring of the brain in the background by LSD. And what's really interesting is, let me just kind of skip ahead here. This is on the Global Brain Connectivity Network, is that these are the changes in default mode network that have been shown on other studies that are hypothesized to underlie the antidepressant effect. We see it with LSD, and we also see it with the 5-HT2A blocker and LSD. It's a little less intense, but it's still there. And the idea with the blocker is we could, and it's actually quite similarly strong. It's just a minor reduction in intensity. But we believe then with the blocker, we would be able to, and you could dose this multiple times then, with this combination as a take-home med, you can still achieve the functional rewiring. So let's look at the actual, what it really looked like here. This is the full map of global brain connectivity of LSD alone. And this is what happens when you have the blocker in LSD.
Wow.
So it's really, really remarkable. No trip.
Really cool.
No trip at all. Intense trip here. Now, they correlate together with an R score of 0.8, which is a very strong correlation. And the two maps on functional network assignment correlate together with an R score of 0.64. So we really are seeing many of those default mode you see here in red, and blue corresponds to a reduction. That reduction in default mode network is present with LSD versus, and these are all placebo subtracted. So placebo's already been removed.
Got it.
With LSD without the trip. So really that made us very excited to then go forward with the fixed dose combination of the psychedelic and the blocker. And we've done that with multiple psychedelics in terms of what we're working on. We're working on LSD, working on psilocybin, working on other psychedelics. So that's really the plan there.
Yeah, that's it was absolutely fascinating to see those results. I guess a question I have for you is, and maybe it's too early to know for sure, but would the idea be for, let's say, neurodegenerative diseases or major depressive disorder, would the idea be that it's a macro dose of LSD, like a high dose, but with that blocker? Mm-hmm. And now that I'm thinking that it would be a macro dose, would it be like a take-home pill, like on a monthly basis potentially?
The exact time frame remains to be determined. It could be a weekly basis, could be 3 times a week, it could be monthly, but that's really the goal, is that you have that pill. It's a full 100 microgram or more LSD, or 25 to 40 mg psilocybin, and you have the blocker. You don't trip, but you get that full dose.
Wow.
So we're really excited about that. And look, this is the primary focus of Terran psychedelic programs, and that's what we built up the infrastructure for. Everything else was just a bonus for increasing patient access. So we said our mission is increasing patient access. We see a lot of ways psychedelics are going to be very expensive and a locked down exclusive market, and we'll break through that exclusivity with all these prodrugs, 505b2s, deuterated forms, and other compounds. So in that sense, the rest of our patent portfolio is for increasing access, and this is the primary psychedelic focus at Terran. But again, we are also committed to those 505b2 plans, and that does still add a lot of potential for other drugs on the market.
Got it. And a question for you here, if you're comfortable with answering.
Sure.
I guess
there are no questions off limits. Here
Beautiful.
You can ask any questions you want.
Beautiful. So I guess maybe playing devil's advocate here. Now we know these the companies that maybe, their business model may be threatened by the 505b2 pathway. What, in your mind, what will they try to do to prevent you from using the 505b2 pathway?
Well, you can't really prevent another company. It's up to the FDA. I mean, they can definitely, we've seen other companies try to build IP moats around psychedelics. And look, we're not there to invalidate any patents that are granted. If someone has a composition of matter and it's granted and it's novel and they're developing it, that's great. We haven't challenged any patents, but the idea, and we believe people should be rewarded for innovation.
Mm-hmm.
Psychedelics, and for the most part, are either off patent, or they're trying to be protected by this whole host of secondary patents, or there are situations where there are large things all still available, deuterated forms, for example, or that haven't been patented and things like that. And in that case, we see that as an opportunity to advocate on behalf of our most important stakeholder, which is the patient. And in that sense, if there's another company that feels threatened in their business model, that's unfortunate, but that's not, our real concern. And so the idea is that, sure, we see criticism sometimes, usually from investors and other companies that come in. It's almost akin to someone saying, it's almost akin to, like, you see someone drowning and you run to help them, and there's a rope right there. So you get the rope out and you're throwing it and you're pulling people in. And then 10 minutes later, somebody shows up and says, oh, why are you saving them? I was on my way. That this was my territory to save people. And I'll take that rope. Thank you. I have my own rope. You don't need to be here. I'll save the patients. And by the way, I'm going to charge everybody at more than it's just like the idea that mental illness is killing people every day. It's damaging lives. And a lot of people like to play the biotech game of exclusivities and we own this and no one else owns this. But we're not talking about a new dating app here. We're not talking about a competitor to, a new TikTok. We're talking about people's lives. Yep. And ultimately, when Terran sees a way that we can break through any of these barriers, get a drug either faster to the patients or bring an alternative form with superior pharmacokinetics or bring a cheaper form, we're going to do it. And, if that messes up the business model of another company, who's typically, well funded by some, multi-billion dollar, venture group, that is just not, our main concern here. Now, we're very collaborative. And we're always happy to collaborate with other companies who want to collaborate. So if there is another company that says, look, we'd like to collaborate with you. Let's do a co-development deal. We see you have this form of the molecule. We have that form of the molecule. We can collaborate together to reach patients faster. We can co-dev. We can support each other in funding to help patients. We are always open to that. And as you've seen, Terran has been very active in collaborations. We've out-licensed compounds from Big Pharma Sanofi. We have acquired compound sets from Concert Pharmaceuticals in an asset purchase. We have out-licensed from Pierre Fabre. And we've licensed academic technologies that have now been one of the first ever FDA clearances with a biomarker for software. So we've done sponsor research for academics, sponsoring clinical studies. We've sponsored multiple times. We've sponsored preclinical studies, and we also provided psychedelics to clinics in Europe and other preclinical researchers. So we're always happy to collaborate, but at the end of the day, if we see an opportunity to exploit technology that we have, we will use that opportunity to reach the patients faster and to help treat these diseases.
Sam, to wrap things up here, what is Terran Biosciences looking forward to the most in the next couple of months, years, maybe?
Well, a couple of big things. We want to get TerraXT into the clinic, and we want to get it on that 505(b)(2) path. That's maybe number one. Idazoxan, we want to get it into that potentially pivotal big schizophrenia trial with our new once daily form.
Yep.
And then I'll just spend a few minutes talking about our software as a medical device, which we're now commercializing. And this is very exciting. So there was a biomarker for dopamine called neuromelanin. It's been around, it was discovered more than 20 years ago. It was actually discovered like 30. But in the last 20 years, there's been hundreds of studies showing it's a great biomarker for dopamine. It could help diagnose Parkinson's disease, Alzheimer's disease, depression. This biomarker, neuromelanin, is a breakdown product for dopamine. All of our brains have dopamine. When it breaks down, it binds to iron in the brain. It forms this compound called neuromelanin. Now, doctors have been calling for this to be part of the standard of care in Parkinson's workup for many years. You can look at all of the different literature where they're saying, it's a great method. It's been used in hundreds of patients. But despite all of that, it was never FDA cleared. The reason why was there was no way to standardize the measurement. So a group and you couldn't standardize it across different MRI scanners. So a group of scientists at Columbia University and also collaboration with Ottawa University solved that. They developed the world's first fully automated algorithms and standardization algorithms to do that. But they developed it in MATLAB, as you would for an academic invention. Now, typically MATLAB inventions, they don't ever get FDA cleared. MATLAB is not an FDA clearable, software. So we actually licensed from Columbia University. And then we spent years also in collaboration with them, but with our own software teams, building out that into a fully FDA approvable software, submitted to the FDA. And at the end of last year, we got the world's first clearance for the measurement and analysis of neuromelanin MRI scans. Wow. It's a cloud-based software, it's it integrates easily with any hospital. You just push a button, it sends it straight to the cloud, sends it back right from the MRI machine, analyzes it in 30 minutes. So we're really excited to bring this to patients, and it demonstrates something that's almost never done, which is taking an academic invention, collaborating with academics who often don't get the to see their inventions actually reach the clinic, and bringing it all the way through the FDA, getting that clearance, and now we're going to bring it to patients where we hope it'll make a big difference. So we're really excited about that as well this year.
That's fantastic. Listen, Sam, thank you so much for coming on the channel and really giving a lot of information to our viewers about what Terran is doing. And I'm excited to see what Terran accomplishes in the coming months, coming years. Listen, everyone, if you enjoyed this content, please like, subscribe, share, and I hope you have a wonderful day.
Thank you. Thanks for having me on.